We're going to be starting chapter 12, the nervous tissue. It's divided into 3 parts, so you can watch this part first and then the other 2. All right, let me minimize myself a little bit. Here. Okay, so the general functions of the nervous system. The nervous system is used for communication and control system. There are 2 control systems in our body, both the nervous system and the endocrine system. You'll get into that one in 169, chapter 17, but the nervous system is one of the ways that we control our body and regulate homeostasis. The nervous system is a lot faster than the endocrine system. The endocrine system uses hormones to regulate and change body processes, whereas the nervous system uses electrochemical signals. So the basic functions of the nervous system are to collect information. There are receptors that detect stimuli and send sensory signals to the spinal cord and brain. So this would be, oh, that's hot, that's bright, that's hot, that's cold, any of those kind of sensory things. I think I said hot twice. It processes and evaluates information. So you have interneurons in your central nervous system that are going to receive that sensory information. They're going to process it. They're going to decide, does something need to be done, what needs to be done, and then they're going to send that information out. They're going to direct that information where it needs to go. And then initiating response to information. So there are effectors that will affect a response based on the stimuli. So the brain and spinal cord will send motor output via nerves to effectors being your muscles or your glands typically. So you have the sensory part where you're collecting information, you have the processing component where you process those stimuli, and then you have the motor component or the effector component where something is actually happening or changing, maybe contracting, doing some vasoconstriction, vasodilation, maybe you're supposed to be secreting more from a gland, whatever that effector response could be. This nervous system can be it can be organized a couple different ways. Your book makes a good point though. There is only one nervous system. All of the nerves throughout your body, you only have one nervous system, even though we do divide it into different components when we talk about it. So the structural organization is central and peripheral nervous system. Your central nervous system is your brain and your spinal cord, and then your peripheral nervous system will be all of your other nerves and your ganglia. There is a third type. There is your enteric nervous system, which is in your digestive system. You'll do that when you do digestive an AMP, again 169. But just to be aware, it's enteric. You can divide that down into the peripheral nervous system because it is not obviously part of the central nervous system. But it is in there, just so you know. But for us, we're just going to deal with the central nervous system and the peripheral nervous system. Note that the peripheral nervous system are where the nerves are. The brain and the spinal cord, they are considered their own things. The nerves and the ganglia are all going to be part of the peripheral system. We can talk about functional organization. So how does it actually work? We can have sensory and motor systems. So the sensory nervous system is the afferent nervous system. It receives sensory information from receptors and transmits it to the central nervous system. So these signals are arriving. Afferent arrives to the central nervous system. The sensory system has 2 components to it though. It has both the somatic and the visceral sensory system. The somatic sensory system is the one that we can actually detect. Right now I have this light shining in my eyes, so I can detect that bright light. It's a little bit warm, so I can detect the warmth. I'm wearing the shirt, so I can feel the sensation of the fibers on me. That's all part of our somatic sensory system, sorry. And it also goes into the proprioceptors. So in our joints, for our muscles, we have what are called proprioceptors that help us to orient ourselves in space. You probably have seen like the DUI test where you have to go and put your finger on the tip of your nose. That's a proprioception test. That's seeing, can you actually orient yourself in space? When you're drunk, you really can't. So I am nice and sober for this class. The visceral sensory system detects stimuli we typically do not perceive. So this is, if you remember, visceral means organs. So this is going to be things like the chemical concentrations in your blood, signals notes say signals from the hearts and the kidneys, whether there needs to be any kind of change in vessel dilation or vessel constriction, any of those, stretching of smooth muscle, that will all be part of your visceral sensory system. The other functional organization is the motor nervous system. This is also called the efferent nervous system. And again, you can think efferent effectors, actually changing a response, as well as efferent exits, the central nervous system. So the sensory nervous system is going to be the afferent coming in towards the CNS, whereas the motor nervous system is going to be efferent going E-way or exiting. This initiates motor output from that central nervous system to the effectors, the muscles or the glands. It also has two divisions, both the somatic and the autonomic motor system. The somatic motor system is going to be our body system. If you remember, I told you when we did muscles, anytime you hear the word somatic, just kind of go towards your skeletomuscular body. That's what that really was referring to. So the somatic motor system is going to be what actually moves and sending signals to your skeletal system. The autonomic nervous system, it's going to send involuntary commands to your heart, your lungs, your organs, all your internal body processes. It is divided into two parts, both the parasympathetic and the sympathetic nervous system, those divisions that we get into in a couple of chapters. But just kind of go ahead and introducing those now. You have both the somatic and the autonomic systems within your motor system. And then within the autonomic, you have the sympathetic and the parasympathetic. This is when I will very much encourage you to write out a flowchart with both the central nervous system, the peripheral nervous system, and then the components of both, as well as the different divisions within each of those systems, just to help you kind of keep things organized. Because once we start talking about, especially getting into the parasympathetic and sympathetic, you don't want to have to take time to remember, okay, is that going to be part of the central nervous system, the peripheral nervous system? Is that sensory? Is it motor? Go ahead and get that organized now. Here is just a basic chart. You could use this one if you want to. I recommend making your own, maybe making one that you could make it much more detailed if you want to. You could do all of your notes. You could write out a flow chart kind of similar to this one, and pretty much all of your notes could be made, for this chapter at least, could be made off of just a simple flow chart, kind of dividing up the two systems. Okay, so now going into actual nerves. So we said, the divisions, the structural and the functional organizations. Now let's actually get into the components of the nervous system, mainly nerves. Those are the functional units of the nervous system. They are a bundle of parallel axons in the peripheral nervous system. Remember, nerves are in the PNS. So a nerve is a bundle of parallel axons. Axons are the long extensions coming from a neural body. So they're bundles of those. So nerve is not a single axon, it's a bundle of axons. They have connective tissue wrappings, just like skeletal muscle does. So this is going to sound pretty familiar to your skeletal muscle stuff. There are three layers of that connective tissue: the epineurium, the perineurium, and the endoneurium. The epineurium is going to be the most outer, the endoneurium is going to be the most inner, and the perineurium is what's going to surround fascicles. Yes, there are fascicles in the nervous system, just like there were fascicles in the muscular system. So the epineurium is on the very outer, the perineurium is going to be containing bundles of fascicles, and then the endoneurium wraps around an individual axon. It's what's actually going to be insulating that one extension. Yes. Nerves are also vascularized, so just like bones, muscles, they have a lot of blood vessels coming to them for both gas and waste exchange. I don't think there's anything in there now. Okay. Yeah. Here is the basic structure of a nerve. You can see this epineurium wrapping all around it. You can see the perineurium kind of defining your fascicles, and this endoneurium wrapping around each one of the axons. There are a couple other things in here that we'll get into in a few slides. Okay, so structural classification of those nerves. So we did the structural classification of the nervous system. Now we're doing structural classification of individual nerves. So just kind of seeing where, trying to keep you oriented to where we're going. We kind of did the nervous systems now. Now we're actually getting into specific nerves. There are cranial nerves and spinal nerves. They follow the rules. So cranial nerves come from your cranium, come from your brain, and spinal nerves extend from the spinal cord. That's not strange. The functional classifications, your sensory nerves contain sensory neurons. Those are going to be sending signals to the CNS. The motor nerves contain motor neurons that are going to be sending signals from the central nervous system. So sensory, remember if I'm drinking something, if I'm feeling something, those sensations are going to my central nervous system, and then the motor neurons are going away from it, sending signals from the central nervous system. There are also mixed nerves, and really most of the nerves that are named that you have heard of are going to be mixed nerves. They contain both sensory and motor neurons. Oops, sorry, didn't do that. Yes, so that's a good point though. Even though mixed nerves, they do contain both sensory and motor components, each of those axons will only transmit a sensory signal or a motor signal. They're not going to be mixing in that way. And then a ganglion. So talking about structures of nerves, a ganglion is a cluster of neuron cell bodies in the peripheral nervous system. So while we said that a nerve is a bundle of axons, a ganglion is basically a cluster of neuron cell bodies. Okay. So some characteristics of neurons. They experience excitability, which is the responsiveness to a stimulus. So if they are stimulated, they will respond in some way, changing their cell potential. They're conductive. They experience conductivity, so they have the ability to propagate an electrical signal, voltage-gated channels along the membrane opening sequentially. So that conductivity, that is the action potential. That's the ability of neurons to send action potentials. This excitability right here, this is what we call a graded potential. So these are two different things. The graded potential, the excitability, causes a stimulus change in the cell's membrane potential in one little local spot. It's in a very, very small region. It doesn't really move around a lot. This is typically going to be, the excitability is typically right at that junction of where a presynaptic and a postsynaptic nerve meet in a synapse when the nerves come together. The excitability portion is only going to be in that small area, and it's just going to send the signal from the presynaptic neuron, where the signal is coming from, to the cell body. So that excitability, it's a very, very small portion of the neuron, or the nerve. Whereas the conductiveness is the ability to move it along its whole axon. So it's going to be ability to actually move that signal, that what was its excitable graded potential, into an action potential that is going to be traveling a distance. So that's kind of one of the two ways that we distinguish those things from being different. If you're kind of wondering what's the difference between excitable and conductive, excitable means, oh yeah, like I'm happy, now I'm excited, but can I transmit that? Can I make someone else excited? That would be conductive. If I could make someone else change, that would be a conductive property. Secretion, release of neurotransmitters in response to conductive activity. Acetylcholine flows out when the nerve action potential comes, and that allows muscles to contract eventually. They experience extreme longevity. By the way, acetylcholine is not the only one, of course, the only neurotransmitter. That's just the one that we've talked about before, so it's the one I wanted to bring up here. Extreme longevity, so neurons can live throughout the person's lifetime. The neurons that you are born with as a fetus can be with you until you are in your 80s and 90s. That's one of their gifts. And they are also amitotic, which is one of their curses. After fetal development, mitotic activity is pretty much completely lost in most neurons. There are some neurons, there are some neuronal stem cells that do remain in the hypothalamus, but that's still being kind of studied. What nerves can do, though, is that they can create new tracks. So while you may not be able to develop more neurons, the neurons that you do have can create new connections between each other. So if you do have, or if you find people that have had serious damage, or they've had some kind of nerve damage, where they've lost the ability to move their arm from a traumatic accident, in some cases, depending on what kind of damage it is, those nerves, through physical therapy, you can actually rehab those nerves. They will build new nerve tracks. They'll create different connections between the nerves and will regrow them. It just takes a long time. It takes about 7 years for that nervous tissue to start to replace it. Not the neurons, but the nervous supportive tissue to replace itself, so that new tracks can be laid. So that's extreme longevity comes with some caveats, in that they can kind of create new connections between each other, but it takes a long time. So getting into the parts of a neuron. So neurons have a cell body, what they call the soma. It has a plasma membrane enclosing its cytoplasm. They call the cytoplasm in neurons the perikaryon specifically. So normal stuff, cytoplasm, plasma membrane. There's a nucleus in the cell body. And the nucleus can initiate some graded potentials that it receives, and it receives others from dendrites. So sometimes it can send out a little graded potential, and it will also receive graded potentials. Those potentials then get conducted to the axon. So dendrites are the parts that are receiving signals. They receive that excitable, that excitability portion of, or characteristic of neurons, comes from the dendrites being able to receive a graded potential, and then the conductive portion is that action potential that gets sent out the axon. They're also, they contain chromatophilic substances, which are their ribosomes. They have free ribosomes that are floating around that are going to absorb stain and change color when you view them. Dendrites, so you have your cell body. You have dendrites, which are short unmyelinated processes branching off the cell body. Myelin is just a lipid sheath coating that we'll talk about in a little bit. But the dendrites that come off of the cell body, those are unmyelinated portions, and they receive input and transfer it to the cell body. So from whatever the presynaptic neuron is, whatever, let's say it's a sensory neuron that's sending information into your central nervous system, it's going to be received, that signal from the sensory neuron will be received by dendrites within the central nervous system. The axon is a long process emanating from the body. So you have the dendrites, which are the short unmyelinated ones. You have the cell body, and then you have one long axon coming off of it, sending a signal out, conducting that signal from the cell body. It makes contact with other neurons, muscle cells, or glands. It attaches to the cell body at the axon hillock, which is just a small portion. So you have your cell body, you have your axon here, and there's a little portion right here that's called the axon hillock, which is a little triangle. Oh, my hands are too low. Which is a little triangle-shaped portion that connects the axon and the cell body. It also, that is the only portion that actually experiences a greater potential on the axon portion. The cytoplasm and the plasma membrane of axons are named a little bit different. We call them the axoplasm for the cytoplasm and the axolemma for the plasma membrane. How it is. The axons end in several axon terminals. Telodendria, these things have lots of different names: synaptic knobs, synaptic terminals, axon terminals. It's all the same thing. The tips of the synaptic knobs, they house the synaptic vesicles, which typically contain neurotransmitters. And axons function to conduct action potentials and then release those transmitters at the synaptic knobs. So that's the whole portion, that's the whole purpose for the axon, is that conductive function of neurons. There's also a cytoskeleton structure to neurons. They have to have some kind of structural features to them. And so you have microfilaments, intermediate filaments, and microtubules, as well as neurofibrils. These neurofilaments get bundled into neurofibrils to provide tensile strength. These will come up when we talk in just a minute when we talk about transport through neurons. So here is the structure of a typical neuron. You can see that you have this axon right here. These little wrappings, this is called myelin. And in between, you have these neurofibril nodes. So what happens is the signal will actually jump down these nodes. This axon hillock region right here, again, that's what attaches the cell body to the axon. You have all of these dendrites that are going to be receiving signals, gets processed in here, and then it gets sent out through the axon. This is an axon collateral, so it's just a little branching off because sometimes your signal isn't going one place, sometimes two things need to receive your signal. Sometimes you got to send a reply all email. And this is just the synaptic knob. This is that terminal portion of it. There's that space in between. Remember the synaptic cleft, because the presynaptic and the postsynaptic neurons don't actually touch each other. There's a space in between. Okay, so neuron transport, how do things move within a neuron? Not just signals, but actual structural components. So axons move material to and from the cell body. If it's going from the cell body, it's called anterograde transport. It's moving newly sensitized materials towards those synaptic knobs. It could be components, vesicles, neurotransmitters, anything. Retrograde transport is coming back to the cell body. So retro, returning to the cell body. It's moving used materials from the axon for breakdown and recycling in the soma, in the cell body. So all the new stuff, it's synthesized and made in the cell body, and then it goes through anterograde transport out to the axon. So you could think antero axon, A and A, whereas retrograde, you're going back to the cell body. This can occur by fast or slow axonal transport. What is that? Well, fast axonal transport occurs at about 400 millimeters per day, which doesn't sound like a lot, but think about neurons. That's a pretty good distance. It involves movement along microtubules, so part of that cytoskeleton structure with those microtubules. So these kind of allow like tracks, almost allow like paths for things to be able to move down. It's powered by motor proteins that split ATP. These motor proteins are not actin or myosin. Actin is actually one of the microfilaments within neurons, but it is not one of the motor proteins that's contributing to axonal transport. What those motor proteins do is they split that ATP to provide the energy for transport. In fast axonal transport, you can have antero or retrograde transport, so either going to or from the cell body. Remember, anterograde is going to be vesicles, organelles, glycoproteins, different necessary new products. Retrograde is going to be used vesicles, and any potentially harmful agents that might get accidentally absorbed into the neuron. Your slow axonal transport is only going to occur from the cell body toward the knob. So it's only going to go in an anterograde fashion. It occurs much slower, about 0.1 to 3 millimeters per day. It results from the flow of the axoplasm. So the flow of the cytoplasm from the cell body to the axon. Remember, once you get into the axon, it's called the axoplasm. It's still cytoplasm in the cell body. It's still a plasma membrane on the cell body. Those things only change when you get to the axon. So once you get to the axon, that flow of the axoplasm towards the synaptic knob is what will just kind of allow things to travel down through it and flow with it. I'm gonna have to keep moving myself around. So structural classification of neurons. They are classified by the number of processes coming off of them, off of the soma. So you have multi, bi, unipolar neurons. Multipolar neurons are going to be your most common type. They have many dendrites, but only one axon. That means that they can receive signals from a lot of different neurons, but they only transmit it out through one. Makes sense. Your bipolar neurons, they have one dendrite and still one axon. There's a limited number of these. Usually, just kind of some of your sensory neurons are kind of like in your eyes. Your unipolar neurons, you have one process that extends from the cell body, and then it splits off into two different directions. So you still have just the one cell body, you have this little unipolar process, and then that process will split off into two. It will split off into the peripheral and the central side. The peripheral process is what's going to be coming from the dendrites, receiving sensory input from your periphery, whereas the central part is going to be going towards the central nervous system to relay that information. You also have what are called anaxonic neurons. These have dendrites but no axons. So these are typically your interneurons. Not all interneurons. Most interneuron are going to be multipolar. But if a neuron is anaxonic, it is an interneuron because it's only going to be receiving that information. We can classify them functionally according to the direction they propagate action potentials. So sensory neurons, we said that they are afferent. They are arriving towards the central nervous system. So the sensory neurons conduct impulses from somatic and visceral receptors to the central nervous system. I always do this, not my heart, but like your brain and your spinal cord. So they come from the periphery to the core. Most of those are going to be unipolar, and a few those will be bipolar in your sensory neurons. Your motor, your efferent neurons, remember those are the ones that are going E-way, exiting the central nervous system. So they're conducting output from the central nervous system to the visceral and somatic effectors. All of those are multipolar. Your motor neurons are all going to be multipolar. And that kind of makes sense. If I need to hold on to something, but maybe it's getting a little hot, but it's also something important that I need to hold on to, I'm going to have competing signals. I'm going to have a sensory neuron signal that says, oh, you need to let go of this. This is hot. Where I might have a higher brain signal that says, no, we have to hold on to this. We can't drop it. So motor neurons have to be able to receive information from multiple sources and then have one effective response. And then interneurons. Most of your neurons are going to be interneurons, 99% of them are, and those are what communicate between sensory and motor neurons. They receive, process, and integrate that information. They communicate it between the two types. Generally, they are going to be multipolar. You don't always have interneurons. Sometimes you can have, like with your short reflexes, you can have a sensory motor, you have a sensory, one interneuron, and then a motor output for quick reflexes. Here is kind of what the general structure of that would look like. This is would be your kind of most simple reflex, whereas you have one sensory input, one interneuron, and then one motor output. So you have an affector, an interneuron, and then an effector. Okay, so where do those neurons actually meet, whether they're sensory or motor or nervous? I'm going to dab just a little bit. Whew, this light is hot. Okay, so the synapse, where they come together. It's two different types. There are chemical and electric. Chemical synapses are much, much more common in humans. We only have a limited number of places that have electrical synapses. In electrical synapse, they are the pre and the postsynaptic neurons are joined by gap junctions, which allow ion flow. Usually you see those in your brain, your eyes, but no real elsewhere for the neurons. These are very fast, though. They're really fast. There's no delay in the synapse transition or transmission in passing that electrical signal, so it is instantaneous that it happens. Most of ours are going to be chemical synapses. You still have that presynaptic neuron that has the axon terminal producing the signal, and you have a postsynaptic neuron receiving that signal. So presynaptic and postsynaptic. Most commonly, that is the dendrites that are receiving the signal. In between is that synaptic cleft, which has the fluid between them. And really, one of the big reasons why we want to have chemical synapses is that you can regulate the response a little bit better. You can amplify the response even more, and it allows for just better control. So the events of the synaptic communication. You have neurotransmitter molecules are released from vesicles of the synaptic knob into the cleft. So if you remember, that action potential will travel down the presynaptic cleft. It will open the voltage-gated calcium channels, allowing all that calcium in the synaptic cleft to flow into the synaptic knob. The calcium will bind to the synaptic vesicle and allow it to release. So the neurotransmitter molecules, so now we'll step into this part. So once all that other stuff happens, the neurotransmitter molecules are released from the vesicles of the synaptic knob into the synaptic cleft. The neurotransmitter will diffuse across the cleft and bind to the postsynaptic receptors. And then that binding of the neurotransmitter initiates some type of postsynaptic potential. And that will be a graded potential. Remember, at the dendrites, you don't have an action potential yet. You have a graded potential, which is just a smaller local change in the plasma membrane charge. The synaptic delay is the time that it takes for all of those events to happen, for the neurotransmitter to be released, for it to diffuse, and for it to bind. Okay, so now we can get into glial cells. These are the other type of cells in the nervous system. These are non-excitable support cells found in the central and peripheral nervous systems. There are lots of different types of 6 different types of glial cells. Glial is just the general term for them. Neuroglia, you'll hear. These are smaller but far outnumbered neurons. I think your book says that in a young adult, they might have 100 million to a billion neurons, but have closer to 1 trillion glial cells. So much, much, much more numerous, many times more numerous. They are capable of mitosis, so they can divide. They protect and nourish neurons. They provide physical scaffolding for the nervous tissue, especially fetal tissue during development. And they're critical for normal functioning at neural synapses. So they provide anatomic support, and they will modify transmission signals just a little bit to be better received. So the first type of glial cells, these are going to be in your central nervous system. These are astrocytes. They have little processes that end in what are called perivascular feet. So look at that word, perivascular. Peri meaning around, and vascular referring to your vessels. So these are going to wrap around your blood vessels and help form this blood-brain barrier here. These are going to be, what this blood-brain barrier does is that it controls which substances have access to the brain. So you think about, you have your brain and you have all the arteries flowing through it. Around those arteries and capillaries are going to be these astrocyte processes, these little feet of these astrocytes that just kind of, if this were like a capillary, the astrocytes are just going to kind of wrap around it in certain ways. And the little spaces kind of between, well, here, let's say it like this. Let's say that my hand is oh, there we go. Where's the angle? There we go. Okay, so let's say the space in between here is where the artery is. The astrocytes will kind of wrap around the artery, and then these spaces between my fingers, this is where it will allow materials to flow through. So even though the capillaries have their own membranes that things have to transport across, the astrocytes provide just a little bit more regulation to make sure that no toxins or harmful substances are getting into the brain, because it is such a crucial part of our body. So astrocytes also help to regulate tissue fluid composition. So the chemical environment around neurons. The book regulating potassium concentration. Remember that potassium is the most important ion for normal neural function, normal membrane potential. So regulating that amount of potassium is very, very important. It will affect your membrane potential and your sodium-potassium pumps. Astrocytes are going to form structural support for nearby neurons. They assist in neuronal development, and they occupy the space of dying neurons. So very important, astrocytes are. Ependymal cells, they line the cavities in the brain and the spinal cord, and they are part of the choroid plexus, which produces CSF, your cerebrospinal fluid. So these are going to be simple cuboidal and simple columnar ciliated cells that are going to line all of the areas, all the cavities in your brain and in your spinal cord, and will help to produce part of that CSF, and then the cilia will help to move it and circulate that CSF. Your microglia, these are immune cells. These are phagocytic immune cells that circulate through your CNS and perform an immune function. They're going to eat infectious agents. And then your oligodendrocytes. These are large cells with slender extensions that are going to wrap around axons and form myelin sheaths. So we'll see that there's another type of cell for the PNS. So these, again, are just CNS cells. And your oligodendrocytes, they just have these projections off of them, these little projections of their plasma membrane that are going to wrap around the axons to help them be insulated. So here you go. You have the sheet. Here are the 4 types. Now, the glial cells of the peripheral nervous system. There are 2 types. You have satellite cells and neurolemmocytes. We had satellite cells. We've seen a different type of satellite cells in, do you remember? Muscle tissue, yeah. The satellite cells are arranged around neuronal cell bodies in a ganglion, and they help to electrically insulate and regulate nutrient exchange. So if you remember that a ganglion is just a grouping of cell bodies, so you have, one of the things is the dorsal root ganglion, just coming off of your spinal cord. So you have just basically a cluster, a grouping of cell bodies, and then around each of those little cell bodies, you'll have these satellite cells that just kind of surround right around their membranes that help to, again, regulate that internal environment and help to insulate them. The neurolemmocytes, you probably have heard more commonly the Schwann cells, but remember, who knows who Schwann was, so we're calling them neurolemmocytes. These are elongated flat cells that ensheath the peripheral nervous system axons with myelin. The myelin sheaths around the axon allow for faster action potential propagation. So that's what the myelin is there for. It's a we'll talk about it in just another slide or two. It has a high lipid content, so that way the axons can be insulated and will allow the signal to jump, what we call the saltatory conduction down the nerve. So yes, here you can see the satellite cells and the neurolemmocytes. So I was saying the ganglion is just a grouping, just a cluster of cell bodies, and then these satellite cells kind of surround all of them. You have, and this is the posterior root ganglion, the dorsal root ganglion of your spinal cord. Here's an interneuron, and then here I will let you tell me what type of neuron that is, but it has these neurolemmocytes. You can see each of these is an individual neurolemmocyte that is wrapping around that axon. In that exposed space, that's the unmyelinated portion. So the yellow that you can still see clearly, that's the unmyelinated portion of the axon. So myelination, what is it, why is it, what's going on? So myelination is the process of wrapping an axon with myelin. Myelin is several layers of membrane of glial cells, several layers of membrane of glial cells. It has a high lipid content that gives it a glossy white appearance and also allows it to be provide a lot of insulation for the axon. The glia are neurolemmocytes in the peripheral nervous system and oligodendrocytes in the central nervous system. So the glia, the cells that are going to be doing myelination for the peripheral, is the neurolemmocytes. For the central are the oligodendrocytes. And again, it's when it says that it's several layers of membrane glial cells. There's a diagram in just a moment. But essentially, what's happening is the cells are just wrapping around and around and around the axon, and they're making these kind of little layers as they go out. So in the peripheral nervous system, the neurolemmocyte encircles the neuron axon and wraps it in layers, forming a myelin sheath. The neurolemmocyte, they can only, so neurolemmocytes in the peripheral nervous system, they can only myelinate a 1 millimeter section of, that's way bigger than a millimeter, by the way. They can only myelinate a 1 millimeter section of one axon at a time. So you have to have thousands and thousands of neurolemmocytes to actually myelinate a full axon. The gaps between them are very normal, and they are crucial for functioning. If the entire axon was myelinated, the signal it would be very slow to transmit. So the gaps between them allow the signal to jump from one to one to one and transmit faster. So here's the myelination of the peripheral nervous system axons. It is different between peripheral and central nervous system. So in the PNS, you can see it starts to, here you have your axon, and here you have your neurolemmocyte. All this is plasma, it's cytoplasm, its nucleus, and the membrane. So what happens is that it starts to wrap around. And if you can kind of think, let's imagine, so it's wrapping in this way. So it's going to be going circles around and around like this. So let's imagine that this portion right here is kind of being stuck. It's going to be laying flat against the axon, and then it's just going to continue to wrap around and around and around until it forms all of these layers called the myelin sheath. And as it does that, it's going to be pushing all of the plasma, all of the cytoplasm in the nucleus further and further to the periphery until you end up with just this thin layer of really compressed cytoplasm and a compressed nucleus, which is called the neurolemma. So again, you can just kind of think about if you're, sorry, my dog decided to eat right now. Winnie, not right now. Go lay down. So you can just kind of think if you have a portion of the plasma membrane that gets kind of stuck and pinned at one point, and then just wraps and wraps and wraps and wraps over and over again around that axon to insulate it. Yes, hi. My dog she wants attention. And then this is what you end up with. So in the PNS, you end up with these this myelin sheath, this incomplete myelin sheath with these neurofibril nodes in between. In the central nervous system, it's a little bit different. One oligodendrocyte can, it still myelinates just a 1 millimeter portion, but it can do multiple axons at multiple spots. There is no neurolemma formed, and part of that is because it retains, you have one cell body that is myelinating these different axons. So as that, it's a similar kind of thing, as it starts to wrap around and around, instead of all of that cytoplasm kind of being pushed to the periphery, not having anywhere to go, it's still connected to the cell body. So all that plasma membrane in the, all that cytoplasm rather, can just be pushed back into the cell body, and then it will go off to a different portion of the axon. Still have the neurofibril nodes. Even in the CNS, you still have those neurofibril nodes. Gotta have them. Okay, and then for unmyelinated axons, because not all axons are going to be myelinated, some of them are unmyelinated. In the peripheral nervous system, the axon just sits in a depressed portion of the neurolemmocyte. So you have your neurolemmocyte here. It has a little depressed opening portion, and the axons are just exposed there. They're not really insulated very well. And then they're enveloped, but again, you can see there's no myelin sheath. They're just exposed. The CNS, the unmyelinated axons are not associated with oligodendrocytes, meaning that there are no oligodendrocytes present to myelinate them.